Nervous system, early development. This sagittal section of the embryo at the beginning of the 3rd week shows the primitive streak, ectoderm, endoderm, and notochordal process. The arrow indicates the ectoderm on the dorsal surface of the embryo. The notochordal process will be transformed into the notochord, which separates from the endoderm. At the end of the 4th week, the notochord induces the overlying ectoderm to proliferate and form the neural plate. The neural plate invaginates in the midline to form a longitudinal neural groove with neural folds on the sides. The neural folds then merge and form a neural tube, which is the primordium of the brain and spinal cord. The notochordal process extends from the primitive node to the oropharyngeal membrane. The formation of the neural tube is clearly shown in this cross-section of the embryo at the end of the 3rd week. Observe the median neural groove and how the neural folds, in blue, merge and fuse to form the neural tube. As the neural tube separates from the surface ectoderm, neuroepithelial cells separate from the neural tube to form the neural crest. The neural crest separates into right and left parts, which are located between the neural tube and surface ectoderm. Neural crest cells migrate widely and differentiate into various specialized cell types. Let us look at the dorsal aspect of an embryo during the early period of somite formation. You can see the neural folds merging in different regions to form the spinal cord and brain. By day 21, there are 3 to 4 somites in the primitive streak, which is regressing. Within the next 2 days, the neural tube is formed with openings at each end, the cranial neuropore, which closes by day 25. The caudal neuropore closes by day 27. The cranial end of the neural tube gives rise to the 3 primary brain vesicles: the prosencephalon or forebrain, mesencephalon or midbrain, and the rhombencephalon or hindbrain. By the 5th week, there are 5 secondary brain vesicles. The prosencephalon subdivides into telencephalon and diencephalon, and the rhombencephalon subdivides into the metencephalon and myelencephalon. From the hindbrain, segmental rhombomeres are formed, and the neural tube continues as the spinal cord. Observe how the neural tube forms in relation to the embryo during the 4th week. Note the embryonic age and the developmental landmarks. By day 21, an elongated neural plate, in deep blue, and a deep midline neural groove can be seen. The thickened cranial region of the neural folds indicate the primordium of the brain. By day 22, somites are formed and sequentially aligned parallel to the neural tube. The paired neural folds have fused, forming a neural tube with brain vesicles at the cranial end. The neural tube extends from the open anterior cranial neuropore to the caudal neuropore, which is also open. By day 25, the embryo appears curved due to folding at its cranial and caudal ends. The rostral neuropore is closing, and before the end of the 4th week, both neuropores are closed. Let us now consider the neural crest cells in a transverse section of the embryo. The animation shows us the neural tube and primary germ layers, as well as neural crest cells. The neural crest cells are extremely versatile and give rise to a wide variety of tissues in different parts of the body. These include the dorsal root ganglia and ganglia of the autonomic nervous system, suprarenal medulla, neurilemmal sheath of peripheral nerves, leptomeninges, pigment cells, and others. Secondary neurulation begins with the formation of the primary neural tube. It occurs in the sacral region, caudal to the posterior neuropore and involves stem cells from the primitive streak and mesodermal tissue. Cavitation in a condensed mass of primordial neural tissue in the embryonic tail bud results in the formation of a secondary neural canal, which connects to the primary neural canal. Finally, the following timeline for the early development of the nervous system and the diagrammatic sketches present a summary of the brain vesicles and their adult derivatives.